Okay, in this video, we're going to look at the role of proteins. Now that we've spent a little bit of time looking at the makeup of proteins, how amino acids are strung together and the different types of proteins out there, or different types of amino acids out there, we want to look at what do proteins actually do. And one of the critical roles is in acting as an enzyme. So what does that mean to be an enzyme? We want to talk a little bit about form and function again. I know we've hit this earlier in the term, but it's a good place to remind you of the significance. What does it mean to denature a protein? And then we'll just do a short little section on nucleic acids as well. Nucleic acids is our fourth famous biomolecule group, and I'm only going to spend a little bit of time on it, not because it's unimportant. It's just ridiculously important, but because we get to some of those themes a little bit later on in the course. Okay, so what do proteins do? What are the functions of proteins? Well, broadly, they're structural support for cells. It helps frame up cells. It's part of the major architecture. They form membrane channels, and right now this may not mean a lot of stuff to you, but really membrane channels allow stuff to get into the cell and stuff to go out of the cell. And so, just like for you, is it important that you have a portal for stuff to get into your body, like through your mouth and through your airways? And is it important to have a portal for things to go out of your body for when you go to the bathroom or when you sweat and things like that? Of course, that's the case. We need these things to interact with the environment. Well, when a cell interacts with the environment, it needs to get stuff from the outside of the cell and bring it in, and it needs to take stuff from the inside of the cell, move it out. And the way we need that is that we just need some doors there. We need some portals. So proteins serve that role. Proteins also carry hydrophobic stuff throughout the body. It probably wouldn't surprise you for me to tell you that your blood is made up of water mostly. So it's a watery environment. It is not a lipid-rich environment. It is a watery environment. And so stuff that is lipid soluble, like cholesterol derivatives, estrogen, testosterone, things like that, they don't have an easy time swimming through the water because remember, they're cholesterol rooted. Remember that they're hydrophobic. So they need a boat to ride on, and proteins serve the function as being boats. Proteins serve as receptor sites for intercellular communication. We'll look at a lot of that later. But then the thing that I want to spend a little bit of time on today in this video is its role as enzymes. All right, so the question then becomes, well, what is an enzyme? Very simply, enzymes are the tools that your body has, your cells have enzymes in order to make chemistry happen. And of course, down the road, we need to have examples of what that chemistry is. We'll start it off very, very soon in terms of some of the chemistry that we use to make ATP, but there's lots of other chemistry that occurs. But my point is that you need enzymes to make them happen. Enzymes, by definition, will lower the activation energy of a reaction, and so how much energy it takes. Part of my brain always had this example where you can think of like let's say an old school middle school dance where all the girls are on one side of the gym and boys are all on one side of the gym. And, if the music's loud enough and there's enough punch and popcorn and things like that, eventually, some girls will cross the gym and boys will cross the gym, and then pairings will happen, so to speak. And again, I know it's probably not like that nowadays. This is in the old days and such, right? But just bear with me. Now, eventually, the people will cross the gym and make connections and such. But if there's an enzyme which can lower the activation energy, meaning it just makes it easier for these reactions to happen. So, hey, Jill, did you know that Bob lives in your neighborhood? Oh, look at that. Oh, do you go to the same pool as I do? Hey, Kim, did you know that you and Billy, both played on the same Little League soccer team or something like that? Oh my gosh, I thought I recognized you. This kind of thing can happen. So the matchmaker is there, and it lowers the barriers to introducing yourself to other people, so to speak. And so by definition, enzymes then lower the activation energy of a reaction, but then are not consumed, meaning that matchmaker makes one pairing, and then he steps away. He's not part of that relationship, and then he makes another pairing and steps away. Is not part of that relationship either, and steps away and does it over and over and over again. So that's what an enzyme does. An enzyme is not consumed in the reaction, but it just makes the reaction way more likely to happen. To say it differently, can chemistry in your body happen without enzymes? And the answer is, yeah, chemistry could happen, but honestly, very slowly, very little, and for all intents and purposes, you'll die without enzymes because you just won't get enough chemistry happening. And so that's why we need enzymes. Okay, so let's just take kind of a visual depiction of an enzyme. This is an enzyme, this is Jack, and this is Jill. These are the two amino acids that are going to get strung together to form a pairing. In this case, it's going to be a dipeptide. So what ends up happening is that these amino acids are going to come and they're going to bind onto this enzyme. And if you look at the shape, you can see how the shape of these amino acids seems to suit this shape right over here. So these are binding sites or active sites within the enzyme. So these two amino acids come together. I like this jolt of electricity. There's no actual jolt of electricity, obviously. It just means that when these things are brought together, it makes it a lot easier for this chemical negotiation, these electron negotiations for chemistry to itself happen. We're seeing here a dehydration synthesis. There's the loss of water, and bam, now we formed a dipeptide. And this enzyme walks away to do it again with another pair of amino acids. Okay, so that's the enzyme. He's not consumed in the reaction. He brought together these two amino acids in a dehydration synthesis, and then he walks away to do it again, and there's my dipeptide. All right. Now, what does this word substrate mean? Substrate it's a word you're going to hear a lot in not just biology, but in science in general. Substrate just means the thing. And so in this context, it means the thing that's being bound together. In other words, some enzymes hook together a monosaccharide with another monosaccharide to form a disaccharide. Okay, in this picture, there are amino acids which form a dipeptide, but I'm saying there are other enzymes that hook together monosaccharide and monosaccharide. In that case, the monosaccharides would be the thing that is joined together, the things that are joined together. Those are the substrates. And here, in this case, it's amino acids. Those are the things that are joined together. So those are the substrates. If I said to you, like, every country in the world has some form of currency. In the US, the substrate of that currency is the dollar. In other parts of the world, the substrate of that currency is the yen or the euro or the whatever. And so substrate really is just a broad term that means the thing of interest, the object of interest that we're talking about. Okay, so there's a good example of an enzyme. What I want you to see then, let's talk about this form and function idea. Proteins themselves have a specific shape. That shouldn't surprise you. We spent time talking about the primary, secondary, tertiary structures of a protein. And what ends up happening when all is said and done, this protein has a certain shape to it, whether it's a tertiary structure or in the other situations where there's two or more that form a quaternary structure. And what I want you to see here is that this shape is not random, it's specific. Meaning that every single time, the hundreds of thousands of copies that I make of this protein, it is shaped exactly the same way every single time. That's just the way it goes. There's precision to the folding here, or the shape of it, or the fancy word would be the conformation, Not confirm, like C-O-N-F-I-R-M, it's conformation, C-O-N-F-O-R-M-A-T-I-O-N, conformation, or the shape of a protein. And so, excuse me, the proteins have a specific shape. The way the protein folds is precise. It is not random. And again, the fancy word that I'm using here is it's the conformation of that protein. It's just the shape of it. So can we agree that if this protein became unfolded or unwound, then it wouldn't be able to carry out its function? Well, visually you should see, yeah, it would seem that these amino acids may not even bind if you disrupted this binding site, or if these two binding sites ended up, like if this thing melted away and this end went to the left, this end went to the right, then they really wouldn't be close enough together for chemistry to happen. So this shape really does matter. Maybe a more intuitive way to look at that is from a different textbook, and this is showing you an enzyme that's shaped like this, all crazy like. This is some substrate, again, the thing that fits on that active site. But if I were to unwind this protein, you see that now that substrate is unable to bind. There's just no way that thing can bind because I've unwound this thing. I've unfolded the thing. So how do you unfold or denature these proteins? In fact, before we do that, can we agree? I guess there's nobody to answer me, so it's rhetorical. But we should agree that if you unfold a protein, you are in trouble. This is bad. You do not want to unfold a protein. Now, if it is intentional and by design, you're unfolding it so you can take it apart to recycle the parts, then it's okay. But you don't want it to become accidentally unfolded. So the question then becomes, how do you unfold or unfold these proteins. Now, the fancy word for that is denature. All denature means, it means the protein itself loses its shape, loses its intended shape. Well, we say that's when a protein denatures. So we do not want it to denature. So what are the common things that can cause a protein to denature? And so generally speaking, the few things out there are high temperature. So when things get too hot, a protein denatures. In fact, this is the albumin in your eggs. If you have a frying egg, the reason it turns white is because the various proteins in there are denaturing. So there's a chemical reaction that happens when we add heat. Now here's what you know. You can't unfry an egg. You know that. Meaning that once you denature a protein, there's no way for us to fold it back up. Now, I know depending on the analogy that you're using, you're thinking, well, what's the big deal? If I take a paper airplane and I fold up a sheet of paper into a paper airplane and I unfold it, what's the big deal? I can refold it. I get that, but that's where some analogies and images fall apart, right? They don't convey exactly what we mean. What I mean is that, look, when we biologically fold up this protein, we do it just right. That if you unravel the dang thing, there is no way to bring it back. You just can't do it. We can't go back to that process, that kind of the origination or birthing process to renew this thing. Okay, so it just doesn't work that way. So the main things that denature a protein are high temperature or changes in pH. Now, the changes in pH, it's going to denature whether you get too acidic or whether you get too alkaline. Remember that our blood pH is 7.4. But so whichever way that you go, if you go too far to low or high pH, you're going to denature proteins. This is what will kill you. So, high temperature. Let's think about this for a second. Where do you experience high temperatures? It's typically when you have a fever. So, if fevers go uncontrolled and they go way too high, then you die. And why do you die? You die because various proteins in critical areas of the brain, for example, denature. And once they denature, once you mess up form, you have messed up function. One more time. Once you mess up form and you denature that protein, you've messed up function. And now, again, you die. This is also why changes in pH are so catastrophic for us. The changes in pH are so catastrophic for us because typically the acidity, and what I mean by that is generally speaking, if your pH goes wrong, as a general rule, you become more acidic. On paper, I get it. You can go either way. You could become more acidic or more alkaline, and they both stink. But in reality, you tend to become more acidic if pH tends to go wrong. We'll talk a lot more about these kind of tendencies and predilections later on or throughout the entire course. But for now, you know, high temperatures or changes in pH can denature. Now, interestingly, cold does not denature proteins. Okay, so it's really just high temperatures. Cold temperatures do not tend to denature proteins. Okay, so that's the idea of denaturing. I've actually said everything about activation energy that I need to. You can take a chemistry class to talk more about this a little bit. The one word I want you to know is catalyst. So what is a catalyst? Catalyst is a thing that speeds up the rate of a reaction without being consumed. So a very simple way to define enzymes, this probably the simplest way to do it, is to say they are biological catalysts. That's what it is. It speeds up the rate of reaction. You may have heard the term catalyst in other contexts. People will say Martin Luther King was a great catalyst for civil rights. And the idea here is that, yes, eventually, at some point, historically, maybe civil rights would have been granted to African American populations, but because of Martin Luther King, that process was accelerated. Well, let's not go too far down that road because it's a crazy time right now, but hopefully you get my meaning when I use that example. All right, so the last thing that I want to talk about, and as I said, only briefly, so we talked about learning objectives 1, 2, 3, 4. The last thing I want to talk about is nucleic acid. This is our fourth biomolecule group, and the nucleic acids are the stuff of genes. When we say genes, with a G in it, we're talking about our DNA, and we'll talk about DNA later, which is why we get to just cover this kind of very briefly. But the DNA is the so-called blueprint for life. Now, nucleic acids themselves are polymers of nucleotides. Right? So there's that word again, polymer. Polysaccharides are polymers of monosaccharides. So polysaccharides might include cellulose and glycogen and starch. Proteins are polymers of amino acids. Well, nucleic acids are polymers of nucleotides. So what we want to do then is we want to talk about nucleotides and look at the building blocks. When you build a nucleotide, And what we're doing here, it's analogous to us building an amino acid, right? What are the rules for building an amino acid? You know those. I don't need to repeat them. So now, what are the rules for building a nucleotide? Well, the rules are you need a 5-carbon sugar. You need a nitrogen-carrying base, a thing that acts as a base, not an acid, but as a base. And then you need a phosphate group. So it looks something like this. So here is my nitrogenous base, my nitrogen-carrying base. In this case, it's adenine. You need a 5-carbon sugar, which is this guy right over here. And then you need a phosphate group, which is PO4. Remember, that's one of our famous functional groups. And this thing right over here happens to be called adenine as a nucleotide. This is adenine. If you want to look at thymine as a nucleotide, and again, I don't need you to memorize these structures. For those of you who have had some DNA stuff before, we're not at a point where we need to memorize adenine, thymine, all those things, and like A matching up with thymine and DNA, etc. I'm just using these as point of example. Here, it looks upside down, but the point is you got that phosphate group, you got that 5-carbon sugar, and you've got a nitrogenous base here in the thymine nucleotide. So these are examples of nucleotides. So again, right now, all I need you to know is that these are the ingredients to build up nucleotides. Nucleotides make up nucleic acids, which is the stuff of DNA, our genetic information. Okay, I'm not going to test you on this in the immediate future, but these are your famous ones, the famous bases involved in DNA: adenine, thymine, guanine, cytosine. All right, so the only other thing that I want to say is one of the most famous nucleotides is ATP. Your next series of lectures is going to look at ATP synthesis. ATP is the substrate. It is the thing. It is the currency for energy in our physiology. Okay, so when we talk about making energy, when you talk about, man, like I slept well and I have, I'm ready to go and all that kind of stuff, a lot of energy, it's generally because you have a whole bunch of ATP available. Okay, take that with a grain of salt. There's a lot of things that can mess with those types of energy levels, but at a cellular level, it means there's plenty of ATP to carry out your functions. In terms of shoving ions in the directions that they want to go, in terms of making muscles contract, in terms of all sorts of stuff like that. ATP is the biological substrate for energy, and we got to talk about its production. So when you look at that, we have adenine right over here, which is your nitrogenous base. We've got a 5-carbon sugar, which is right over here, and here we've got a phosphate group. We happen to have 2 more, which is why it's called ATP, adenosine triphosphate. These two things together form adenosine. If you add one phosphate group, it's adenosine monophosphate. If you add another one, it's called adenosine diphosphate. If you add a third one, it's called adenosine triphosphate. Okay, so all I need you to know for now is that one of the most famous nucleotides is ATP. To recap, we've covered all of these learning objectives, and that'll wrap it up.